So, I've got a lot on my mind when it comes to cell membrane potentials and ion channel integrity when it comes to the development of type 1 diabetes. Let me tell you why. So, there was a paper in 2019 that found out that verapamil, which is a calcium channel blocker typically used for things like heart disease, and it had protective effects on beta cells and the production of insulin in newly diagnosed type 1s. And you may be wondering yourself, well, why would a calcium channel blocker typically used for heart disease have anything to do with diabetes? And it has everything to do with how insulin is actually released in the body. And it turns out calcium is incredibly important for the release of insulin. So in healthy beta cells, glucose metabolism leads to an increase in the ATP to ADP ratio, which closes ATP-sensitive potassium channels and depolarizes the cell membrane. In response to this depolarization, this opens the voltage-dependent calcium channels, allowing these calcium channels to now enter the cell. When calcium ions enter beta cells, that is the signal to release insulin. And now there are things to address at every step of that process I just talked about, whether it be ATP production and mitochondrial health in the beta cells, whether it be sodium and potassium balance, allowing for that cell membrane to be able to be depolarized, or if it's calcium ion channels being opened and closed properly via the voltage of the cell so that the insulin can get out. But one of the things that piqued my interest was viral influences on calcium dynamics. So I'm going to read a quick little bulleted summary of a paper called Host Calcium Channels and Pumps in Viral Infections. So this is a paper that looked at how viral infections impacted host calcium management. So calcium ions are vital for virus entry, viral replication, maturation, and release in host cells. Viruses alter host cell calcium homeostasis by manipulating the channels and pumps, benefiting their lifestyle. Okay, why does this matter even more to people with diabetes? The endoplasmic reticulum is one of the most important organelles in beta cells. And this is important because the endoplasmic reticulum is responsible, among other things, for protein folding. And if the endoplasmic reticulum, isn't working properly, it can be misfolding proteins, i.e., not making your insulin properly. And in states where there is a high rate of misfolding of proteins in beta cells, that is like they're trying to make insulin proteins, but it's not going well, all of those misfolded proteins accumulate within the endoplasmic reticulum and cause oxidative stress. And this can trigger the unfolded protein response, or UPR, which aims to restore normal function, but if prolonged or severe, can lead to beta cell dysfunction or apoptosis, which is cell death. Chronic ER stress is implicated in the development of diabetes as it impairs insulin production and contributes to beta cell loss. Calcium dynamics and our body's ability to properly utilize calcium is imperative for proper insulin release and beta cell health. Viruses are very well known for hijacking their host's calcium dynamics. And when they do this, it sometimes comes at the cost of the cell that they have infected. And thus, drugs that are focusing on reestablishing proper calcium balance are having success in saving beta cells and reviving insulin production. So you have to be wondering aloud, what are the ways in which we can properly reestablish calcium dynamics for beta cells? And what are the best ways that we can address the factors that disrupted that calcium balance to begin with? And of all the steps or phases in insulin release, mitochondrial health, sodium potassium balance, calcium influx, is there one that is more important than the other? What do you think? This kind of has me a little bit excited.